Hi everyone, my name is Lu and I'm a PhD student from Karolinska Institute. I'm here to present to you a method we have developed for the isoform quantification of single-cell RNA sequencing data. There has been an exponential scaling of single-cell RNA-seq data over the past 2 decades, and numerous sequencing technologies have been developed, with the most used being the droplet-based and the full-length RNA-seq techniques. The full length, as the name suggests, enables the sequencing of whole-length mRNA molecules, whereas a droplet-based method, only part of the mRNA molecule near either its 5' or 3' end can be captured. A good example is illustrated in this diagram. SMART-seq2, which uses a full-length single-cell RNA-seq protocol, follows a pretty uniform read coverage. Whereas on the other hand, 10x, which uses the droplet-based technique, has a non-uniform distribution peaked near the end of the gene body. Several methods have been developed in recent years to estimate isoform expression for the full-length method, but so far, no method has been developed to address the prime bias problem. To fill in the gap, we developed Scasa, a method to estimate isoform expression for droplet-based single-cell RNA-seq technologies. The whole process of estimating transcript abundance revolves around the use of alternating EM algorithm. Standard isoform-level quantification tools such as Callisto or Salmon uses similar model, and the difference is that the X matrix is assumed to be known. In contrast, both X and beta are treated as unknown parameters in Scasa. On the macro level, Scasa consists of 3 parts. First is the mapping and pre-quantification preparation step in the sample data. Scasa utilizes existing pseudo-aligners such as Callisto bus tools or Elwin to carry on mapping, and the output will be a recount equivalence class matrix as shown on the figure. We treat each cell in the data as an independent event, and for each of them, vector y is computed by summarizing all sequence reads for each equivalence class in the cell. We assume that y follows a Poisson distribution with mean and variance mu, such that mu follows the bilinear model x times a beta coefficient. X is the design matrix that summarizes exon sharing between isoforms and is unknown. Beta is the transcript abundance that we wanted to find out, and which is also unknown. So before we proceed on to the EM algorithm, we need to construct an initial X matrix for the model. Polyester package in R is utilized by Scasa to produce expression counts that are representative of an actual 10x single-cell RNA-seq data. And at the same time, Scasa also models the prime bias brought about by the droplet-based technology. We then obtain a set of equivalence class just like the sample data's equivalence class. As a process of optimization, we break down the X matrix into independent X matrix transcript clusters, such that all isoforms that belong to the overlapping equivalence classes are included in one cluster. So now, given X and Y, we could estimate the transcript abundance of every equivalence class, which is the beta parameter here, by a summation of all the mu's generated from the transcript clusters. And this is done independently for each cell. Then use the just computed values of the transcript abundance beta, given beta and y, at the second step, which is the maximization step of the AEM algorithm. We compute a most likely estimate for X. So the 2 steps iterates until convergence. Based on the simulated data with ground truth, we compare the estimated counts of Scasa with other quantification tools. And as you can see from the first row, in terms of isoform quantification, Scasa outperforms all other existing methods. Even when summing up the transcript counts onto gene level counts, Scasa also outperforms all gene count estimation methods and shares similar performance with Star Solo at gene level. We tested Scasa on a bone marrow data and found a new CD14 monocyte cluster, as you can see from here, which diffused back into the main CD14 monocyte cluster when gene expression quantification were being used. Top isoform of this cluster came from the same gene, thyroid, which showed distinct differential expression patterns across its isoform parallels. And their expressions observed to be very different within the CD14 monocyte cells. At gene level, differential expression effects were being masked. And as a result, the cells from this subgroup share more similar phenotype at gene level and were diffused back into the main CD14 monocyte population, as you can see from here. This work has been published, and Scasa is available on GitHub for download and use. I would like to thank everyone who has contributed to the success of this project, and thank you everyone for listening. Goodbye.